afternoon and welcome to today's EMSL Learn webinar. My name is Genoa Blankenship, and we're excited to have you here so that we can discuss capabilities available for your biological research and how you can access them through two upcoming calls for research proposals. The next EMSL Learn webinar will be held next Wednesday, same time, noon Pacific Standard Time, December 7th, where we're going to be discussing available computing focused capabilities. We'll have more information about these upcoming calls available on our social media channels and also through our email subscription. If you would like to stay updated, we encourage you to follow along. Today, we have Scott Baker up first. He's going to be walking through those two upcoming calls for proposals, including timelines and important information that you're going to need to know about. James Evans will follow. He's going to be demonstrating how to use nanotomography for generating 3D representations of cells. Sarai Williams will also be speaking. She's going to be providing details on using EMSL's popular small-scale automated sample analysis workflows, including nanoPOTs for proteomic analysis. And Sankar Krishnamoorthy will round out our presentations by discussing how you can use activity-based probes for functional characterization of proteins. We encourage you to stick around for the whole hour. The last 15 minutes will be devoted to your questions. So we encourage you to post questions in the chat, and our speakers will answer them at the end, 12:45. And with that, I'm going to toss it over to Scott Baker to get us going.
Thank you, Genoa. And thank you all for joining this webinar. I want to thank also our comms team, Genoa and team, for all the organization that they've done. So I'm Scott Baker. I'm the science area lead for functional and systems biology. I'm excited to tell you that our calls will be opening up soon. And there we go. Okay, so we have two calls opening up. The first is the large-scale EMSL research call, or LSR call, and these are for projects that last up to 2 years. And we have focuses in our 3 science areas, which are environmental transformations and interactions. The webinar for that was a couple weeks ago. Functional and systems biology, which is today, and then next week, as Genoa mentioned, computing, analytics, and modeling will have a webinar. This call opens December 14th with letters of intent due in February and full proposals due March 23rd. Oh, whoops, now I went too far. The ficus proposal is an exciting partnership that was originally EMSL and the DOE Joint Genome Institute. And this is also for access up to 2 years, and it connects researchers with both EMSL and JGI through a single proposal mechanism. And then in addition, for this upcoming proposal call, we have partners at Neon, Biosands, and the APS. So these are all these can all be accessed through the ficus mechanism. So it's not only EMSL and JGI, but these other user facilities. It's been really exciting to see the proposals that come in here, and the focus areas will be coming out in the next few weeks. It opens January 9th, and with a letter of intent due in March and a full proposal in May. Let's see if I can advance the slide correctly this time. Yes. For more information about the proposal calls, you have a QR code, websites, and an email address. So multiple ways to contact us. Next up, I'm going to introduce James Evans, who will be talking to you about nanotomography. Take it away, James.
Thank you, Scott. While we're trying to get control, perfect. All right, so thank you for the introduction. And in the next few slides, I will discuss a new X-ray nanotomography capability at EMSL that enables 30 to 100 nanometer resolution of 3D visualization for samples composed of biological, organic, and also inorganic materials. Now, this new capability was motivated by the realization that PNNL and EMSL had a gap in compatible sample thicknesses for nanoscale and label-free ultrastructural 3D imaging of cells and tissues that hinders correlative imaging. In particular, PNNL has imaging systems that directly visualize 3D ultrastructure without labeling at resolutions greater than 1 micron and less than 10 nanometers. This means that we were unable to visualize whole living organisms intact and label-free with nanoscale spatial resolution to fully understand their internal organization, composition, and cellular changes in response to environmental perturbations. For example, what is happening morphologically to thylakoids or chloroplasts when photosynthetic organisms go through a diel cycle. From a graphical perspective, consider this image at the top, which is a confocal stimulated Raman scattering image. This shows us a living cell and allows visualization of particular components, but not the whole cell context. Even super resolution fluorescence can only see labeled fluorophores. However, we can do cryotransmission electron microscopy at the angstrom to few nanometer spatial resolution to visualize the native cell context and see fine details in carboxysomes and thylakoids. But to get these images, we must use an ion beam to mill the sample into a thin cross-section, which removes the majority of the whole cell context. In this case, those middle images of carboxysomes were derived from a lamella cross-section that was only 1/20th of the normal cell diameter. Now, previously, to address this, we visited synchrotron facilities and took advantage of a soft X-ray beamline available at the advanced light source to provide 30 nanometer spatial resolution of intact fungi that were 10 microns thick. Our goal was for this particular project and the new capability that I'm talking about here was to develop a laboratory-based soft X-ray nanotomography system here at EMSL for analyzing biology in native environments to complement both the capabilities existing at PNNL and EMSL, as well as the other DOE synchrotron facilities. Now, with PNNL funds, we wound up being able to acquire and commission a laboratory-based soft X-ray nanotomography capability to fill this 3D imaging gap. We partnered with Sigray to develop the first ever nanofast biolambda instrument capable of imaging biological samples in a lab-based setting, using two different soft X-ray beam energies and allowing both absorption and phase contrast. In the bottom right, you can see an example of a diatom, and in the upper left, upper left, you can see the first demonstration of 30 nanometer resolution imaging on a test sample with only a 1-minute exposure. These two images proved that the instrument was functional, and separately, a built-in optical microscopy beam path allows for direct targeting of specific fields of view for downstream correlative imaging. Now, our final commissioning experiment sought to compare the lab-based system that I'm describing here with what we previously recorded at the advanced light source. We were able to get comparative images of algal cells containing significant amounts of lipid droplets. The bright white round features are the lipid droplet, while the darker gray region is the chloroplast in these cells. Now, this particular image on the right is of a dried sample, but they show the same features as cryogenically hydrated samples that were previously imaged at the advanced light source. And while we don't need the results to be exactly the same, the hope was to help minimize impact and access to synchrotron facilities via sample screening beforehand, while also allowing direct multimodal analysis with other capabilities. With that demonstration in hand, I'm happy to announce the soft X-ray nanotomography capability is now available for EMSL users and can be used in a number of ways especially for linking 3D ultrastructure from the nanoscale to the mesoscale for biological systems, organic polymers, environmental and particulate samples, and even energy materials. And furthermore, before ending, I do want to point out that this system is intended to work in concert with other capabilities at EMSL to enable hierarchical tomography and imaging across scales. To learn more, please visit the EMSL website or contact me, or visit the EMSL YouTube channel where you can get more details about synergistic approaches like the high-resolution cryo-electron microscopy and cell-free expression webinars that were done in the last year. And with this, I'd like to hand it over to my colleague, Sarai, who will talk about automated nanosampling techniques for small-scale and single-cell proteomics, LC-MS analysis. Thank you.
Thank you, James. Hello all, I'm Sarah Williams. I'm a chemist here in EMSL, and I'm here to talk to you about some automated single cell and small scale proteomic techniques available for potential users. Whoops, I went too far. Okay, so what is single cell proteomics? Well, we have this analogy of a fruit smoothie. And here the fruit smoothie is represented by like bulk scale proteomics, where you have like a mixture of many different types of fruit. And if you were to ID the context of this smoothie, you would get an average response, but it's most likely that you are going to miss some features. Single cell proteomics can be represented with these isolated pieces of fruit here. And if you were given something like a fruit salad, you could sort through the fruit salad and you could be able to tell what's there and like how much. And then for spatially resolved proteomics, we have this the representation, we have this beautiful chart. Every fruit here is in its own space, very distinct from each other. So choosing a pipeline that's best suited for your experiment will be determined based on the initial sample size. So if you're looking for an average response of the cellular diversity that's expressed in your biological sample, you can use traditional proteomics, which takes generally a large amount of single cells in the order of millions of cells or hundreds of micrograms of starting material. And then over here you have nanoproteomics, this is where you're looking for abundance and dynamics that are featured in the cell-to-cell interactions which are happening in your biological sample. And that's something you want to look for. You would need a starting sample under 1 microgram. For instance, we have micropots here. So if you have a sample that's less than 500 nanograms, you could inject 20 microliters of that starting material. And for nanopots, it's a much smaller sample size. So we have an injection of 200 nanoliters. And for that, we would need something much smaller, something less than 500 nanograms. And as you can see, like as your sample size decreases, so do the challenges. And challenges increase for mass spec proteomics. One major challenge is cell size. So here we have 4 different types of cells. We have E. coli, we have fungal cells, we have plant cells, we have mammalian cells. So the smaller the cell size, the smaller the sample volume, and therefore that will affect how much peptide is available in that sample volume. So for E. coli, we cannot amplify signal like transcriptomics can do. But what we have is a starting material. Therefore, we do not have a technology that's sensitive enough to quantify peptides in a single E. coli cell with the starting volume of 1 femtoliter. We do have like well-established pipeline for mammalian cells. However, the cell size for mammalian cells are very different. You have the HeLa cell, which is starting materials around 200 picograms, and then you have something much smaller like a lung tissue, which is 20 picograms. So that starting material really makes a huge difference. And now we have another challenge on top of that challenge. So we already expressed that sample loss is a huge issue, and sample material is a huge issue. So with normal traditional pipelines, we have these like surface areas where we are losing peptides to these non-specific binding sites. Each star here represents sample loss that is connected to the surfaces of these the surfaces of these vessels which hold our samples. And then each micropipette here represents peptides or proteins that we are losing due to non-specific binding of pipette tips during aspiration and transferring. nanoPOTs proposes to address these challenges first by reducing the sample surface area down to like 1 millimeter, 2.2 millimeters. Here's a video. We have, this is the second way. Here we have our automated robotic system, which is dispensing all our reagents in one spot. This allows us to avoid sample loss through excess pipetting and surfaces. Okay, moreover, despite reducing the sample surface area, we are increasing the reaction volume. If you look here in this table here, like using an Eppendorf tube method, your reaction volume is around 100 microliters, but using nanoPOTs, we're injecting like 200 nanoliters, right? That's the most those wells can hold. Second, like the surface contact area has been reduced by 99.6%, making reactions like much more sensitive. This also increases our digestion kinetics up to 500 times. And because this is an automated system, our sample prep is very sensitive, very robust, and higher throughput. So we, as nanoPOTs, we established ourselves using mammalian cells, but now we have capabilities for plant tissues using 2 cell isolation techniques. For single cell, you can use Selenium 1 or fluorescent cell sorting, and this is like represented here with protoplasts. For spatial single cell proteomics, we have laser capture microdissection, and here are 3 examples of what types of tissues we can use for cell sorting. Here is an example of a well-established single cell pipeline using popular plant tissues. This protocol was published in a methods paper if you'd like to read more about it. So during this pipeline, we took two types of leaf samples and 2 types of root samples. And we were able to isolate these, the cells from here using cryosectioning and laser microdissection. If you look at the bottom part of the slide, you can see that you can see this area where you can see an area where the targeted tissue is directly catapulted into one of these nanoPOTs wells. That way we were able to quantify what is being expressed in that cell at that time. Next, the samples were digested and processed using the nanoPOTs proteomics pipeline. And here you can see on this side, the before and after of the 3 different types of tissues. Okay, so these red bars, the red color represents vascular tissues, and the green color represents calcified tissues. And these bars go up or down based on upregulation or downregulation. And then over here with our PCA plot, we can see that the proteins being expressed are forming nice clusters, different and distinct from each other. For a single cell example, we have an example from a user, Gary Stacy. He's very interested in nitrogen fixation through soybean nodules interacting with bacteria. So we were able to use fluorescent cell sorting to dispense 400 protoplasts per well using 3 different cell types. Then we're using our nanoPOTs platform, we're able to send it through the pipeline to yield some really impressive preliminary results. So here you can see that in the PCA plot that the cells they're all clustering very different. The cell types are very different from each other. And then if you look in our volcano plot, you can see that peptides are being expressed differently based on the type of, based on the control versus the treated. And then you can see all these pathways over here. So that's really all I have to say about nanoPOTs and small cell proteomics. We hope to see you working with us in the future. Thank you. And here is Sankar.
Hello, everyone. I'm Sankar Krishnamoorthy. I'm a chemist here at EMSL. Today, I'm going to be talking about activity-based probes, how you can use them to functionally profile some of the biological system you're working with. I'll introduce you to 2 types of activity-based probes, and then I'll touch upon substrate activity screening approach for novel protein function and cell function characterization. All right, so basically we are trying to manipulate the biological system to do our bidding towards, for example, biological material production or biofuel production. In order to successfully do that, we have to have a better understanding of the biological system that we are working with. So we get a lot of information from genomics, transcriptomics, and proteomics. However, there is a disconnect there because those provide a relative abundance of, for example, the proteins. But since the proteins are the closest in dictating the phenotype, you want to have better understanding of the protein function at a given time or at a given environment. So So the proteins actually play a vital role in cellular life. They have a variety of functions. I have listed some of these functions, but these functions are carried out by these proteins through biomolecular interactions or small molecule interactions. So for the activity-based probe, we take advantage of these small molecule protein interactions. You can think of an enzyme that does a biomolecule transformation where it takes a substrate and convert it into something else. So there is an interaction of that substrate to that enzyme. So similarly, there are a lot of small molecules that protein interacts with, but arguably you can actually find one small molecule that functionally affects these proteins, whether it shuts down the function or it increases the functional activity of the enzyme. So we take this protein small molecule affinity and incorporate 2 additional elements to make these activity-based probes. For example, the black box here actually is the affinity element from the protein small molecule interaction. And we incorporate a reactive group on that element. And we also incorporate a reporter group. So the reactive group allows these probes to bind the protein irreversibly, and the reporter group we use to analyze and get more information about the protein. So typically, the probe development involves understanding of the targets and understanding of the protein small molecule interaction. And we will design a probe and synthesize it to get the probe in pure form. And then we validate it using either pure protein, a non-pure protein target, or cells or organisms with the protein of interest. So once we validate the probe, they are actually ready to be used in activity-based protein profiling pipeline. So the sample actually is either part of live cell or lysis or a protein extract. So we take these probes and incubate it with these samples, and the probe binds the protein target, and we use click chemistry-based approaches to attach a biotin, which then can be used with streptavidin to enrich for the function, the probe-bound proteins. Then that we digest it with trypsin and analyze it with mass spectrometry. So from the mass spectrometry analysis, we can actually identify the protein targets that actually reacted with this activity-based probes. Since the probes are designed based on a certain type of protein small molecule interaction, you can actually say that these proteins have that particular function. You can also take the probe labeled proteins and couple it with a fluorophore and use it for STH space analysis, where we can visualize these protein bands by doing fluorescent gel analysis. You will get molecular weight information. You can also do in-gel digestion and do mass spec analysis of that protein band. In addition, you can transfer the protein onto a membrane to do Western blot analysis if you have antibody available for that particular protein target. So if your proteome is part of a whole cell, we can actually take those cells and do fluorescent imaging to locate where these targets are. We can also use fluorescence activated cell sorting to enrich for the target-rich population, which then you can use for functional analysis, or you can do sequencing. You can even do proteomics with what nanoPOTs that Sarai introduced to you. So in the next slide is one specific example where we developed this probe that targets one particular type of enzyme involved in immune system evasion phenotype of pathogenic streptococci. So the data that you see is from flow cytometry, where we did an initial validation with target species with a negative control. Here, these are from monoculture labeling of with the probes. You can see the target organisms are labeled compared to the negative control. Similarly, we can work with overexpression system. You can see the negative control, the overexpressing E. coli get labeled more than the negative control. The third chart that you see is mixed cultural labeling, where we have a negative control and the target organism mixed together and then label it with the probe and analyze it with flow cytometry. You can see there's a separate population of probe labeled target organism. So the second type of probe that my slide is not advancing, excuse me. Okay, there we go. So the second type of probe that I want to introduce you is the activity-based phylogenic probes, where the recognizing moiety is attached with a dye. In this state, it's actually off. You don't see the fluorescence. When it is acted upon by an enzyme, the bond gets cleaved and the fluorophore or the dye becomes active, that you can actually see it on the, for example, in a plate reader assay where as the activity goes, the fluorescence keeps increasing. So this type of probes can be used in different types of samples. And these are the types that you can actually commercially access. Or we can actually design a specific probe for your enzyme proteins of interest. And we can also do live cell fluorescence imaging by specifically designing these probes. We can actually look at where the enzyme activity is using some of the fluorescence microscopy techniques. So we have a broad spectrum probe, where if you're looking at a big enzyme family, for example, like a glycosidases, or we can actually also design probe to look at one specific enzyme in a bigger family of enzyme. So I introduce you to two types of probes where you actually need the substrate information. If you don't have that information, that's where we have this substrate activity screening. We developed this large library of like a synthetic compounds attached with a fluorophore that is very similar to the second type of probe that I talked about. And then we take this library and screen it with the either the protein lysates or live cells, for example. So when there is an activity, you see fluorescent increase, and you can pick those two compounds. But you can pick the compounds that actually have higher activity and modify it into an activity-based probe that covalently binds to the target protein. And then you can enrich that and see the proteins that are actually responsible for this activity. Another thing that you can do is to take those high acting probes and use it in different biological samples to identify which sample actually has this unique activity that you're looking for. So we developed one such a library for amide hydrolases with 150 compounds, and screened against one soil microbe to identify two probes from that library, and modified them into an activity-based probe that we use to enrich proteins and do proteomics analysis. So the pie chart that you see here is actually from that sample. The point that I want to make here is we identified different types of hydrolases, including amide hydrolases, but we also identified about 41% of proteins that did not have any annotation. So now you can go and say that these proteins likely have some type of hydrolase or amide hydrolase activity. So that concludes my presentation. I just wanted to summarize that we have these activity-based probes that you can use for biochemical functional characterization of your protein of interest or in biological system, in addition to gaining other information. We already have probes that you can actually use through these upcoming calls. We can also design a particular type of activity-based probes depending on the system that you want to investigate. Through the substrate activity screening library that we developed is actually available for screening biological samples. And I also want to say that EMSL is an excellent place because we have access to expertise in chemical synthesis, chemical biology, in addition to having access to all sophisticated instruments for this type of studies. Thank you.
Great. Thank you to all four of our presenters. We are going to enter the Q&A portion of today's webinar. So would ask all of the speakers to turn their cameras back on and we'll get rolling on the questions we've received. And I would encourage you, if you have questions, to go ahead and put them in the chat and we will be checking those. The first question is from Ann Summers. The question is, reduction step risks losing metal adducts, natural and toxics. What reductants can you use and can you leave out for that step altogether?
Is that a nanoPOTs question?
You will know better than I will.
Okay, so the work I was talking about today is bottom-up proteomics. However, we have done native proteomics and top-down proteomics. But for bottom-up proteomics, yeah, so if the salt isn't covalently bonding, we will definitely use that during our reduction. And we do use DDT and TCEP for our reduction agents. Hope that answers your question.
Anne actually had an additional question. For living cell analysis, can you do longitudinal time series experiments and do label compounds engender an idiosyncratic effect themselves, for example, off target? At what molecules or cell volume do you use?
Is that nanoPOTs? Because we do not do living cell analysis. However, people in the building do. And I recommend for you to speak to Chris Anderton about that because they do stuff like that. Okay. I think we can put a link in the chat.
Yes, absolutely. We'll list his email address for you, Anne, in the chat. Next question is for Scott Baker. What is the best strategy for developing a successful EMSL or FICUS user proposal?
Okay, that's a question we get often, and I would say the most important thing to do is to reach out to all of us at EMSL. We're here to help you put together the most successful proposals. So when we find that the best proposals are those where people have worked with our integrated research platform leads, our instrument scientists, and I'm one of the. We have all the email addresses and contact information here. You can always email me, and I can funnel somebody to the correct technical expert. But I think that's been, the biggest part of people's success is when they work with us to help craft the proposals. So we're here to help.
Great. Okay, let's see here. Sankar, do organisms survive the probe processing when you isolate using FACS?
All right, so that's a good question. So I think there's the second half of Ann Summers' question also involves like labeling. So it depends on the probe type and the organism that we're working with. But we can actually design probes that does not toxic affect the organism. We have done, for example, the example that I showed with streptococci actually allows us to isolate live cells after incubating with the probes. So the probe volume, the probe amount and the cell volume, usually we do those labeling with, for example, 50 microliter sample with like 0.5 to 1 OD worth of cells. And the probe concentration varies depending on how effective those probes are. We screen, for example, from 0 to 1 micromolar.
All right, thank you. Anne's hand is up, so I am going to unmute her, and just in case there was something that she could add to her question. So Anne, go ahead and unmute if you can. Feel free to ask your question.
Oh, sure. Thanks. Well, I did have a question that I wanted to ask those two separate speakers that were my two separate targets questions, but I think that's been answered. So I thank you for, I've forgotten to remove my hand. So sorry. Thank you very much.
That's okay. No problem. Osa, I'll ask your question. And if we don't answer, you can leave your hand up. So in using the probe and or flow for, I hope that's what they meant, for enzyme assay, how do you address the variability issue? Is the assay result reproducible?
Yes, so that's a good question. So we usually do replicates of all the samples. So you usually get an error bar, for example. They are most reproducible.
Okay, good to know. Okay, Sarai, another question for you. In what capability can EMSL users be involved with the cell isolation in the nanoPOTs pipeline?
It's a really good question. I think the answer to that has several parts and it depends on the experiment. Now, if you are interested in spatial proteomics, we have two options for you. So some facilities have their own cryosectioning and their own LCM. And so for users like that, we send our nano well chips to them, and then they sort the samples for us. They send to me for processing. Now, if they do not have an LCM, they're welcome to come here and work with Bo Chrysler, or we have other staff that do that, and we could use the LCM to sort the cells with them, and then they can be with us while we are doing the nanoPOTs pipeline. And then for fluorescent cell sorting, we have single cell fluorescent cell sorting, so you can use fluorescent cell sorting and celling in one to like dispense your wells into the chips. Now, if you have that also, we can also send chips to you for that. So, yeah. Thanks.
Very good. Let's see here. Sarai, another question for you. What kind of sample prep is used for proteomics, and to what extent do users need to prep their samples in order to do that, to do nanoPOTs proteomics at EMSL?
Okay, so we do our own sample prep, which is isolated using our nanoPOTs robot. Okay, so the sample prep will be done here, and the users can be with us while we do the sample prep, but we don't recommend them doing their own sample prep. However, if they are isolating their own cells, they could send a frozen tissue, and we can do the cryosectioning for them, and we could do the LCM and our bottom-up pipeline, which I showed in the presentation. And if you have more questions about our sample prep, I can send our previous manuscripts.
Very good, great. We did have a question in the chat about this recording, and in general, these recordings are available on our website in the EMSL Learn section, usually about a week after the webinar. So last week's on environmental research and capabilities at EMSL will be available later today. I'm going to do a last call for questions. We do have 15 more minutes of question time if anyone has any additional questions. I'll give it another minute or two. Looking in the chat to see if there's any
Waving a hand.
Oh, you are. Thank you. I'm glad that you go ahead, Anne.
Okay, this came up actually for, I think this is really for Scott. I noticed on the webpage that there's description of the sort of things you can apply for throughout the year kind of thing. And these larger proposals are limited to just a certain time. And I think I heard you say in the earlier previous talk that if you want to get in for the year, you got to get in now at this particular call, because otherwise there won't be another one for a couple of years. But they are also on that webpage. There's a call. There's for the small proposals, the kind of pilot project things. They're all in green, as if almost any month you could receive a request for a very short proposal. Could you clarify that for me?
Sure. So the LSR and FICUS are yearly, once a year, open once a year, as is the exploratory call. So we have 3 different main proposals open that have a single deadline, and each has its own review panel. What you're probably referring to for the one that's got green for all 12 months is our limited scope proposal, which is kind of a really small experiment that if you're thinking about like a figure for a paper and you need an experiment, or maybe you want to just see how something works. We have kind of a limited number of limited scope proposals. So that's something where you might contact me and I can put you in touch with the appropriate integrated research platform lead, and we can see if there's an experiment that makes sense to do via the limited scope. I think obviously the best, the most high depth access is through our LSR and FICUS calls, and then the exploratory. But we're always looking to bring in new users in whatever way we can, given our current resources. So there are limits, but probably your best bet, yours and anybody else on the line, is to contact me, and I can move you to whoever can help you best.
Yeah, this arises from a conversation that came from the AI meeting, so I'm good there, and thank you very, very much for that clarification.
No problem.
Yep.
Right. I am not seeing any additional questions, but I just want to remind you that all of the speakers are available via email, and as are the integrated research platform leads and the instrument owners if you have additional questions. And I would invite you to join us next week, noon Pacific Standard Time, for our last webinar in this particular series, looking at computing capabilities for the 2 upcoming open proposal calls. And be sure to keep an eye on our website for the details. And we just thank you for joining us today. Have a great day.